All right. So I believe the screen is visible to everybody.
Yes, ma'am, it is visible.
Okay. So, this is where we left off last time, you will recall. And it is basically about the biogeochemical cycles that we are talking about. And in this, there are several elemental cycles which I wanted to discuss with you in order to apprise you of the fact that things like carbon or nitrogen, etc., they get recycled in the environment and we need to maintain a certain balance. Now, as we had seen in the case of carbon, that if there is excess of carbon dioxide in the nature, then it is going to lead to, for example, global warming. And there are different kind of biochemical activities which keep on going in the environment where carbon dioxide is fixed up, that is by plants or by certain photosynthetic bacteria, into organic matter. Organic matter is again respired through microbial activities to give rise to carbon dioxide and water. So what doesn't seem to be very conspicuous is that there is a delicate balance of carbon which is operating through these activities. The last leg of that carbon cycle was oxidation of C1 compounds. So I have already discussed it with you. I will not repeat it again. But in the question answer, if you come back and say that there is something which is not clear to you, we can always come back. So I will start out with nitrogen cycle now. And just as carbon, the nitrogen is also cycled in the environment. Okay, so I'm going to read out certain things and then I will explain very briefly what nitrogen cycle is all about and also what are the different parts of it which you need to understand, and then what is its practical significance. So as it says here, as opposed to carbon, elements such as nitrogen, sulfur and iron, these are taken up generally in the form of mineral salts. Okay, now why we say that? Because carbon dioxide is fixed by the photosynthetic organisms, and that is the way carbon is only in the gaseous form that it is assimilated into the biosynthetic into the biological systems. Okay, on the other hand, when we say elements like nitrogen, sulfur and iron, these are taken up as salts. Okay, so that means the nitrogen will be taken up in the salt in the form of salts like nitrate salt. It could be sodium nitrate, it could be some other nitrate, or it could be as an ammonium salt like ammonium chloride or ammonium nitrate. And there is only a very small part of a biological system which are called as the nitrogen fixing bacteria, which are able to fix molecular nitrogen. That is, they can take up nitrogen in the gaseous form as nitrogen and fix it into ammonia. Okay, since that leg is extremely small, the rest of the nitrogen is usually taken up as nitrates or as ammonium salts. That's the reason why we say that these elements are taken up as mineral salts. Similarly, when you look at sulfur, sulfur will be generally taken up as a sulfate. Okay, so you have like gypsum, you have calcium sulfate or potassium sulfate. That is the way sulfur is taken. You don't have any microorganisms which can take up sulfur dioxide gas or H2S gas and it can assimilate it. That is the difference. Similarly, iron will be certainly taken up as a salt because it doesn't exist in the gaseous form anyway. And we will see that these elemental cycles, they will operate through oxidative and reductive pathways. Okay, and I will explain to you in a while as to what we mean by that. So nitrogen, for example, can exist in different oxidation states. For example, in ammonium, if you try to find out what is the oxidation state of nitrogen, you will see, you have 4 hydrogen, which is 4, and it is a cation plus. So the valence of nitrogen here, the oxidation state is -3. Okay. Similarly, if you look at the oxidation state of nitrogen in a nitrate, it is actually +5. Now, unlike carbon, carbon was always, when you look at carbon dioxide and something, it is always existing in a fixed tetravalent form. But here what you have, it can exist from -3 to a +5 oxidation state. So that is the difference. Similarly, sulfur also transits between different oxidation states in different kind of compounds. Now, nitrogen is important because it makes up almost 12% of the cell dry weight. Like carbon is a predominant element, similarly nitrogen also is a predominant element, and it needs to be taken up. And once it is taken up in the biomass in such large quantities, then you need to release it back into the environment. Otherwise, the cycle is not going to be completed. So all the nitrogen in the environment, whether you take it up as a nitrate salt, or whether you take up as an ammonium salt, or whether you fix molecular nitrogen in the form of ammonia and fix it in the biomass, eventually it has to be recycled back. All that nitrogen should be returned back into the environment in the form of nitrogen gas. Okay, so is this clear to everybody as to why we emphasize so much on this recycling?
Yes, ma'am.
All right, so now just as the global carbon cycle is undergoing a change, global nitrogen cycle is also undergoing a change. Why? Because of excessive use of nitrogen fertilizers, burning of the fossil fuels, land usage changes, they all impact the nitrogen cycle. Now, needless to tell you that when you use nitrogen fertilizers, obviously you are adding potassium nitrate or ammonium salts in the form of fertilizers. You are adding too much of nitrogen into the soil, and through rain and through water, much of this ammonium and nitrate goes into the water bodies where you have excess of nitrogen, which causes algal bloom. And we had already talked about what is the disadvantage of algal bloom. So too much of nitrogen leads to eutrophication, and there is too many algae are growing, and that adversely impacts, for example, the ecosystems. Fossil fuel burning. Why? Well, because if you burn the fossil fuel, you are going to release gases which are oxides of nitrogen, like nitric oxide or nitrogen dioxide. They are all released into the environment. Okay, so too much of nitrogen is entering, the nitrogen cycle is disturbed. When you burn fossil fuel, again, oxides of sulfur, sulfur dioxide, all the gases are entering into the environment. Too much of this, you are again disbalancing now the sulfur cycle. So that is the reason why it is of concern to us, because we are bothering the nitrogen cycle, we are bothering the sulfur cycle. Iron, because of mining activities. Because iron is a constituent of steel or construction activity. There's a great deal of mining activity going on, as a result of which too much of these ores and the mine tailings, whatever is left behind of the ore after the iron is extracted, all those are called as the mine tailings, and they also cause too much of pollution in the form of addition of iron. Okay, so this is something which therefore needs to be kept in mind. And very briefly, so when you talk about the nitrogen cycle, please remember that eventually all this nitrogen, is in the form of molecular nitrogen, which is in the environment. And where does it come from? So constant volcanic activity in the environment are responsible, and Earth's volcanic activity are responsible for release of molecular nitrogen. That is the source of nitrogen gas in the environment due to volcanic activity in the Earth's core. So as the first part of the nitrogen cycle, nitrogen has to be fixed, and that is carried out by what is called as the nitrogen fixing bacteria. So these are the bacteria which can take up molecular nitrogen, they combine it with hydrogen, and they make ammonia. Okay, so just as carbon dioxide is fixed into organic carbon in the form of starch, the same way the atmospheric nitrogen can be fixed up by these bacteria, which are called as the nitrogen fixing bacteria, to make ammonia. Okay, so now this nitrogen is trapped in the form of ammonia, and this ammonia can enter into biomass. That is called as ammonia assimilation. So ammonia will be now incorporated into all the nitrogen containing compounds like nucleic acids, or you have like purines, pyrimidines, and other nitrogen containing compounds in the cell are going to benefit from this ammonia which is taken up by the cell. So it becomes a part of the organic matter, and eventually there are these nitrifying bacteria which will convert back this not really ammonia in the form of gas, but in the presence of there is high acidity where too many protons are there. So it becomes ammonium, it gets converted to nitrate, and then back into nitrogen. So that is how the nitrogen returns back into the environment. Okay, and so this is, very, very briefly, it is just to give you an idea of how nitrogen cycle is going to operate. So this is one particular part. The other part is that proteins, etc., are ingested by all microorganisms. Okay, so in the form of amino acids, etc., proteins, enzymes are essential. So when these are excreted, it is in the form of ammonia. Okay, so that ammonia again is available, and it also undergoes cyclic reactions, and eventually, it ends up as molecular nitrogen. So this is what the basic concept is, but we are going to examine it as to what are the different legs of it. So is this clear to everybody?
Yes,
So
ma'am.
other
Ma'am,
students,
one thing like you told that the source of nitrogen is the volcanic activity. So like, is it is this the case when the nitrogen was not in atmosphere? So uh, by that process, it came into atmosphere, or what you are trying to say here?
yeah, certainly, because gradually, uh, whenever now, even today, you say that nitrogen makes up a major part of the Earth's atmosphere. So the question is, where does this gas come from? Okay, and this gas basically comes from the volcanic activity which occurs in the Earth's core. That is the basic source of molecular nitrogen. Okay, so that is where the nitrogen is. And of course, through biological forms, nitrogen is first of all assimilated by the nitrogen fixing bacteria, or in the form of nitrates or ammonium salts. It can be taken up by other microbes, etc. But eventually, whether it is the molecular nitrogen, whether it is protein which you are taking, or whether it is the nitrate salts, whether it is the ammonium salts, etc., they all come from atmospheric nitrogen. Okay, that's why I think it's very
Yes.
important to understand. They all come from atmospheric nitrogen. Okay, that needs to be cycled into these compounds, and then eventually this nitrogen has to be returned back into the environment. And today, like I said, that the global nitrogen cycle is undergoing a change because of use of excess of nitrogen fertilizers, fossil fuel burning. We are disturbing this delicate balance of nitrogen, and that's where we have to be aware of what needs to be done so that the nitrogen cycle is not balanced in the environment. So just as with the carbon cycle, we talked about very briefly, we can't spend too much of time discussing so much in detail, but again, conceptually, because it is an important cycle, I will let you know about what are the basic nitrogen reservoirs in nature. That is whether you talk about atmosphere or land or ocean, then how this nitrogen is going to be fixed, that is by microorganisms, where nitrogen is converted into ammonia. And you can think it's very, very similar in concept to CO2 fixation, where photosynthetic bacteria will take up carbon dioxide, they will fix it into organic carbon, like starch or starch. Starch is predominantly the product of photosynthesis. And then you will have uh, nitrogen assimilation. In case the nitrogen fixation, only very small group of microorganisms are able to fix atmospheric nitrogen. All other microbes will take up nitrogen in the form of these salts, which are nitrate, ammonium salts, etc. So we will look at it. And the final step is denitrification. That is, denitrification means removal of nitrogen in the form of nitrogen gas. So in this process, we will see how this nitrogen enters back into the atmosphere in the form of nitrogen gas. Okay, so very briefly, so we will just learn only to appreciate, you don't have to remember the numbers, but yes, you have to know that where all is nitrogen, and what is the nitrogen, what is the form of the nitrogen in which it is going to be actively recycled back. So these are the nitrogen reservoirs, metric tons, and whether these are actively cycled the same way as we discussed about the carbon cycle. So in the atmosphere, the source of nitrogen is the nitrogen gas. Okay, and as it says here, that nitrogen is continuously released into the environment from the volcanic and hydrothermal eruptions, and is one of the major reservoirs, which is 3.9 times 10 to the 15 metric tons of nitrogen. Okay, and there is only a small part of it which is fixed by microbes. So overall, the question is that nitrogen actively recycled? The answer is no. Okay, although it's a huge reservoir of nitrogen, but there are only very, very small groups of microorganisms which are called as the nitrogen fixing bacteria, which are able to take up this atmospheric nitrogen. Okay, and we will look at it as to what is the process, what is the importance of this fixation. In ocean, the nitrogen is of course present in biomass. So, all your biological organisms, etc., they serve as a huge reservoir of this, and they keep dying, decaying, etc., decomposing. So yes, although it's a small reservoir in comparison to this molecular nitrogen, but it is actively recycled. Okay, similarly, organic matters which are in there, because of the, biomass is generally kind of living cells, but this is organic matter which comes, proteins, or any other kind of lipid molecules, or lipids, carbohydrates, etc. These are all called as organics, actively recycled. Then there are the soluble salts. Then there is dissolved nitrogen also, because nitrogen which is in the atmosphere exists in equilibrium with what is in ocean, a large reservoir, but then it's not actively recycled. So what you are dependent on for nitrogen recycling are principally these particular sources, which are all constituents of the biomass. Okay, land biota means all living things. Okay, so this is also actively recycled. Then Earth's crust has so much large reservoir, but then it's not really exchangeable with the environment. So now,
Ma'am, I have a question.
yeah.
So, ma'am, you said that these only the rhizobiums, and there are very a few numbers of this bacteria which can fix the nitrogen. So, like in a jungle, there are there are trees, or lots of trees, and they are big trees, and you see that. Like all this biomass on the earth, they all have this nitrogen. So are they getting all this nitrogen from this bacteria, from this fixed by this bacteria only, or is there any other way? If not, how can they fix so much amount of
Biomass.
nitrogen?
So through the roots they are taking nitrates. Through the roots they are taking ammonium salts. These are not plants which live in association with the nitrogen fixing bacteria. All plants, all trees which you see outside, they take up nitrogen in the form of nitrate, ammonium salts. They don't have any association except for a handful of plants, like your legume plants, for example. They are the only ones in the roots of which you have the nitrogen fixing bacteria. So whenever you have crops, etc., which are growing, like you have wheat crop or you have rice crop, etc., you will see that in between they always plant legume crops in there, so that, because for rice and for wheat you have to add fertilizers, because there is not enough nitrate salts or ammonium salts in the soil in order for them to be building a biomass. So when you are trying to grow rice and wheat and so many other food crops, they are all dependent on external fertilizers you are adding. And that's the reason why there has been a practice of crop rotation. That is, when, for example, wheat is harvested, then there is some kind of a period in between before the rice is sown. In between, they try to put those plants which are leguminous plants which are able to fix the nitrogen. So there is crop rotation. So you will put a crop, for example, legume crop, in which the roots of which will have an association of nitrogen fixing bacteria, and they are able to fix up atmospheric nitrogen. They make ammonia, and that ammonium is released into the environment now. So the soil again becomes enriched with the ammonium salt. So does that answer your question?
Yes, ma'am. But take for example, I am planting a tree here somewhere, and then this is a big tree. Let's say it is a people tree, a big people tree. So it will have like nitrogen, some percentage of like 10 or 20% of will
Biomass. Yeah.
consist nitrogen. So where this nitrogen comes?
Oh, it comes only
Not.
through the soil in the form of nitrates, ammonia, because trees like that have grown over 50, 60, 70 years. So the root system also is not very stationary. a very tall tree is going to have roots which are going to extend over 0.5 kilometer down in the ground. So they capture these nitrate salts which are constantly occurring. So there is a lot of microbial activity. When there is dead organic matter, leaves, etc., which are falling, there is a microbial activity, proteins, leaves are decomposed, proteins are broken down by enzymes, proteases into these amino acids, etc. Then amino acids are also denitrified. Nitrate is returning back to the soil. That is their only source of nitrogen. They don't fix atmospheric nitrogen.
Yes, ma'am. Yes, ma'am. Understood now. Understood.
Okay.
Okay, ma'am.
Okay. All right. So now, so therefore, the nitrogen fixation is the first leg which I'm going to discuss with you. And nitrogen fixation, for example, now, there is how many metric tons of nitrogen do we really fix per year? Okay. So natural nitrogen fixation occurs on land. It also occurs in the aquatic systems and also through lightning. Okay, so what do we mean by lightning? That is, nitrogen is present in the environment. Hydrogen is also present in the environment. And through whenever there is a lightning or something, there is enough energy for this nitrogen gas to combine with hydrogen to give rise to ammonia gas. Okay, and that is also a very large source whereby nitrogen gets fixed, 10 to the 7 metric tons per year. Okay, then anthropogenic, that means the man-made nitrogen fixation. This is through managed farming. What do we mean by that? Is crop rotation. Okay, so we deliberately, for example, in our farming practices, we deliberately have crops which are going to fix the nitrogen. That is called as managed farming. And fertilizer manufacture, through Haber's process, that in any fertilizer plant, you have the nitrogen, the gaseous nitrogen is made to combine with hydrogen to give rise to ammonia. And that is how the fertilizers are manufactured. So is that clear? So these are also the ways that we are able to fix up the nitrogen. Anthropogenic fixed nitrogen mobilization. So, what are the ways that the nitrogens are mobilized then? Okay, so through fossil fuel burning, you are returning nitrogen in the form of nitrogen dioxide, the gases, etc. You burn the biomass, or wetland draining, land clearing, etc. I'm not very clear about what are the agricultural practices, but these are also some of the practices through which the nitrogen is mobilized. Mobilized, that means if it is in the fixed form, then it is made to come out in the form of either nitrogen dioxide or nitric oxide gas, etc. Like in fossil fuel burning, nitric oxide is released, nitrogen dioxide is released. Okay, so 2/3 of the nitrogen is fixed by microbes. So when you look at this number, it is relatively large. In all this, if you just calculate how much of nitrogen is fixed, then you will see 2/3 of the nitrogen is fixed by microbe and 1/3 is from the fertilizer production. Now, cost of fertilizer production is extremely high and it involves consumption of fossil fuels. So crop rotation is recommended, like I said, because this way, there are crops like soybean, etc., which are rotated in order to fix atmospheric nitrogen. Okay, so extremely, because eventually the source of nitrogen on Earth is going to be only the molecular nitrogen, the nitrogen gas. So now let me quickly review. So is this clear to everybody?
Yes, ma'am.
All right, so other students also, if you have any doubts, yes, or if you feel, there is something where you need to have clarification, so please pay attention to it. You can listen to lectures later on, but the learning is not the same as you do sitting in a classroom. Okay, so now in the biological nitrogen cycle, here is an overview. All right, now the figure seems very complicated, but then we will try to understand it in different parts. Each one of them is going to be explained in the next few slides. So when you look at the nitrogen cycle, we start with the nitrogen gas. Okay, and as I said, the nitrogen has to be fixed into ammonia. All right, but since ammonia, when it is released, for example, in the soil by these bacteria, all right, because of the natural texture of the soil, because of the change, because of the pH of the soil, there are too many protons. The soil is generally acidic. Okay, as a result of which the ammonia gets converted into ammonium ion. So ammonia NH3 plus a proton. So now it is available as ammonium ion. So this process of fixation is called as dinitrogen fixation. And you see the number 4 is written here. This only indicates what enzyme is responsible for it. Okay, so we look at on this, for example, in the legend, it says 4 is nitrogenase. So those handful of bacteria which are able to fix up atmospheric nitrogen, they have the enzyme nitrogenase. And that is why they are able to convert nitrogen. I will give you again a detailed slide on this, that how nitrogen can combine with hydrogen to give rise to ammonia. We don't show it as ammonia because in the acidic pH, it gets converted into an ammonium ion. Okay. Now the second leg, which you see here, the process which is called as nitrification, is the process whereby this ammonium is converted into nitrate. Okay, so what do we mean by nitrification? Nitrification is a process whereby the ammonium ions are converted into nitrate ions. And there are several groups of it's not a single microbe, but there are groups of microorganisms which carry out this reaction and they have the necessary enzymes. For example, 11 is called as now ammonia monooxygenase. Okay, so you can see monooxygenase because you look at ammonia NH3, you add another oxygen to it, so it becomes this hydroxylamine. Okay, so those group of microorganisms, they have this particular enzyme, and some other microorganisms or the same group of microorganism could have another enzyme which is hydroxylamine oxidase, which converts it into nitrite and then to nitrate. Okay, so this leg or this process is called as nitrification. And then we also have a process called as nitrate assimilation, because as I said that most of the living forms, they take up nitrogen in the form of either ammonia or they take it in the form of nitrate. So nitrate is first converted into ammonium, then it is assimilated into the biological systems. Okay, the last leg of the cycle is called as denitrification. So by denitrification means removal of nitrogen completely and making it available in the atmosphere as nitrogen gas. Okay, so try to understand what these different terms are. So this is how the nitrogen cycle operates in the environment. All right, so what you see here, for example, down below, is that nitrogen, it undergoes all these different molecules which you see. The nitrogen exists in different oxidation states. Beginning with, if you look at what is the oxidation state of nitrogen, in ammonium, it is actually -3. So what you see down here is the state of oxygen, is the state of oxidation of nitrogen in these different compounds. Okay, so in hydroxylamine, for example, the state of nitrogen, oxidation state is -1. In nitrogen gas, it is 0. In N2O, it is +1. In nitric oxide, it is +2. So I hope you're able to match all of these. Okay, so similarly in nitrate, the state, oxidation state of nitrogen is +5. All right, so what you see on this particular scale is the nitrogen scale, which is in all of these compounds which you are looking at. So please try to pay attention to it. And as you go from -3 to +5, obviously these are all oxidative pathways, because you are adding electrons and eventually you are going to -2, your removal of electrons, that's the oxidation process. So you go from -3, -2, -1, so on and so forth. Okay, and here you are adding electrons, so therefore these are reductive pathways. So nitrate assimilation is a reductive pathway, nitrification is an oxidative pathway. So is that clear to everybody? So this is a very, very key table. It's a very key figure. Try to understand as to what all these different things mean. Remember, you're expected to remember these enzymes, because we will see that eventually when you talk about application of the nitrogen cycle, one of the major applications of the cycle is in the wastewater treatment plant. Because when you treat the wastewaters, in the domestic waste, for example, there is a lot of urea which is coming in, or in the domestic waste, you have, for example, the different proteins, the food materials which you put into the dustbin eventually get hydrolyzed through microbial activity into proteins. Okay, so that means by protein, what you mean? A lot of this ammonium, and eventually in the wastewater treatment plant, all this nitrogen, also needs to be mineralized. You want to remove the nitrogen from this ammonium containing plants. So activities like nitrification, denitrification are extremely important in a wastewater treatment plant. Otherwise, there is no way that the nitrogen which is now trapped in the food material, it is going to be recovered back. Otherwise, the food material, if it is not processed properly, all that nitrogen is going to remain trapped as dead organic matter, or it will just get accumulated as cell biomass, which is in the activated sludge process. So do you understand? So can somebody please repeat what I said just now? So please, one of you, can you just come forward and repeat what I said about the importance of the nitrogen cycle? Kumar Kaushik, Abhinav Thapa, Kartikeya, Ashu, are you there? So where have you all disappeared? Harshit? Ratnesh, are you there?
Yes, ma'am, I am here. I could not hear clearly, ma'am, last word.
Kartikeya, are you there? Divakar, so people just log in and they go off, mind their own work, whatever they are doing at home. So anybody else in the class you want to come forward? Divakar? So Ratnesh, was that point clear about, why nitrogen cycle is important and why it is important to know what are the different activities which are going on?
Yes, ma'am. If this processes, we know about this process, we can use this processes for in like wastewater treatment plants and etc. Using this
Because
enzymes.
because because nitrogen recovery, when you are treating the wastes, okay, so there is a lot of food matter, and eventually it is hydrolyzed into proteins and amino acids. So today, there is a lot of thrust in recovering this nitrogen from the food, from these amino acids, so that the nitrogen, denitrification can be carried out and this trapped nitrogen can be returned back into the environment. It's like recovery of an essential mineral if you are able to carry out denitrification, nitrification as a part of the wastewater treatment plant. So that's why it's important to know. And many of these microbial activities today are going to be coupled in the wastewater treatment plant so that nitrogen, you are able to recover nitrogen. So anyway, let's continue. And because you see, even I was looking at the minor exams and the performance is so dismal by the student because people are not listening and they just look at visually the PPTs without understanding anything eventually. So anyway, let's continue. And so in this biological nitrogen cycle, I will just finish up this particular slide to say that, these are the important legs here which people need to understand. And over and above, what you see here is that something which I have added here is a process which is called as ammonia oxidation. All right. And it has been found that in nature, all of these processes, you see the conversion of ammonia into nitrate, it is an oxidative pathway. There are a number of oxidative steps which are going on, which are a part of this nitrogen cycle. But over and above that, ammonia can also be oxidized under anaerobic condition. Okay, by and what do we mean by that? Because oxidation doesn't mean always participation of molecular oxygen. Oxidation also means removal of electrons. And if there is an electron acceptor other than molecular oxygen, still you have oxidation which is going on. And that is called as the ammonium oxidation. Here, nitrite comes as an electron acceptor and nitrogen gas is formed. And this process of ammonia oxidation occurs under anaerobic condition, and it is extremely important in the environment to return back nitrogen into environment. So we will come back and we will see how processes like ammonium oxidation are being integrated into the wastewater treatment plant, and these are supposed to be much better and more efficient compared to aerobic processes. So the first part, which I have just projected here, is that of dinitrogen or molecular nitrogen fixation. So in the last slide, for example, we have this nitrogen fixation. So each one of these, which I have shown here, is going to be explained in the following slides. So you have nitrogen fixation, and now in nitrogen fixing systems in the environment, okay, although you have heard about nitrogen fixation occurring in the plant roots, which is called as a symbiotic nitrogen fixation, where both plants and bacteria are going to participate together. But that's not the only way the nitrogen is fixed. It can also be fixed by free living bacteria. Okay, and there are, as it shows here, there are at least 100 different bacteria which have been identified in the soil, which are capable of fixing atmospheric nitrogen. But how much do they really contribute? It's about 1 to 2 kilogram nitrogen per hectare of land per year. So they make up only a very small percentage of nitrogen fixation. And then we have something which is called as rhizosphere. That means there are bacteria which are associated with the soil, which is called as the rhizosphere association. Then there is a cyanobacteria moss combination. These are bacteria plus moss, which give you about 30 to 40 kilogram nitrogen. But you see the largest share is between the rhizobium and the leguminous plant. They are able to fix up large amount of nitrogen. And that's why we attach so much importance to this, because they make up very, very large chunk of the nitrogen fixation process. So briefly, what exactly does the process involve? So here what we have shown on the top is a bacterial cell. So, eventually, like soybean is a crop where in its roots, what you see that there are certain nodules, swellings, okay, in which these nitrogen fixing bacteria are going to reside. Okay, so this is where these are going to reside, the bacteria. So what kind of association occurs here is explained here. So this is a typical bacterial cell, and this is the cell of the plant, which is the root cell. Like you see here, it says plant cell, and then you see here it is, for example, it is a bacterial cell up here. Now these bacteria, for example, they have they have the enzyme which you see here as a nitrogenase enzyme. So nitrogen is taken up from the environment, and these are sulfur, which is actually it's a nutrient which is taken up from the soil as a nutrient. So these are transporters which are located along the membrane. Molybdenum, which is a cofactor for the enzyme, which is nitrogenase. Okay, now nitrogen will combine with hydrogen to give rise to ammonia. That's what is shown here. Okay, and what it means here is that oxygen inhibits the process of this nitrogen fixation. If there is too much of oxygen, it also inhibits. So this is basically in a small compartment in this corner of the cell. You have anaerobic environment which is created, highly reductive environment, in which the nitrogenase is going to bring about reduction of molecular nitrogen. Okay. So you see, so this ammonia, like is shown, for example, in this cell, that the nitrogen.
Ma'am,
So
here
this
in this process, from where the hydrogen comes, if oxygen inhibits this process, then definitely it should not come from H2O. So from where this
not
hydrogen comes?
it is in the gaseous form because hydrogen is present in the environment. So hydrogen also enters as a gas inside and it combines with nitrogen to give rise to ammonia. Okay, the only purpose of showing oxygen here is to let you know that oxygen is an inhibitor of nitrogenase enzyme. And nitrogenase is a very complex enzyme. The entire process involves reduction, electrons, etc. So all of these that you see here, for example, are the various genes. This shows for nitrogen fixation, NIF gene cluster, etc. So the gene products of these, they code for various proteins and they make the complex nitrogenase, which is able to fix the molecular nitrogen into ammonia. So ammonia comes out, and in the soil, the ammonia will combine with proton to give rise to ammonium ions. And how does the plant benefit? So the plants will take up these ammonium ions inside and they will assimilate this ammonia. Okay, so this ammonia, in other words, so in our biological systems, there are enzymes like this is called as glutamine synthetase. So there are glutamic acid is an acid, but then if it accepts an amino group, it gets converted into glutamine. Both are amino acids, you will recall. Okay, glutamic acid has a, of course, it has an NH2 group, which is the terminal NH2 group, but it has a carboxylic acid. So that COOH, OH gets replaced by an amino group right here. So it makes glutamine. And this glutamine is the principal donor of nitrogen for nitrogen containing compounds. And then it's responsible for plant growth. Okay, so this is the way the plants are going to benefit, that this nitrogen becomes available to them. The question is that how does this bacteria benefit? Then various acids of the TCA cycle are synthesized, and many of these acids, they are transporters which take them into the soil. So the bacteria can directly take up malate and other organic acids, flavonoids, which are compounds which are, which are part of various coenzymes, etc. So they are secreted in the soil and they are taken up by the bacteria. So it's a win-win situation. The bacteria also benefits from the presence, from its association with the plant. Okay, so that's why, because both of them are benefiting from each other. So the growth of the bacteria is much faster and it is able to fix up more nitrogen, and you're able to fix up a large amount of nitrogen here. Okay, so this is the first leg as to how nitrogen is fixed. Then the second part, you will remember in that slide where we were discussing about the nitrogen cycle. So once ammonium is taken up, then how does ammonia get assimilated? Okay, so very quickly, so we talked about ammonium assimilation here. So now we are going to examine how this ammonia, which comes from nitrogen fixation, gets assimilated in the form of amino acids like glutamine, etc., or other nitrogen containing compounds in the biological systems. Okay, so in order to understand that, let's examine very quickly. So there are two principal reactions by which ammonia is assimilated. Now, under the condition where the ammonium ions are very high in the soil, okay, then they are taken up immediately by the cell, and in the presence of enzyme which is called as glutamate dehydrogenase, this is alpha ketoglutarate, which you see here. Ammonia gets incorporated here and you make glutamate. So what you see here, for example, is that at this keto group, for example, the ammonia comes in and this is how glutamate is formed. And the enzyme which is responsible is glutamate dehydrogenase. So you are expected to know both of these pathways. That is how ammonia is assimilated. This is one way which occurs under very high ammonia concentration. But if the ammonia concentration is very low, then ammonia is taken up by glutamate. It's converted into glutamine. Okay, and then this is donated now in the form of glutamate. So these are, glutamine synthetase enzyme and glutamate synthase. So just make sure that, all these three enzymes are very, very different from each other. The first process occurs under high ammonia concentration, and this process occurs under low ammonia concentration. Okay, so this is the way ammonia gets assimilated. All right, now we may talk about mineralization. That is, how does really from these biological systems, how does ammonia get out? That is ammonia mineralization. That is the reverse of the process. Okay, so reversal of these reactions can easily occur and ammonia can just come out. That is mineralization. How do you really get it out of the proteins and biological systems as ammonia, which is excreted out as a waste material? Now, in the higher life forms, in the higher life forms, ammonia is excreted as urea, which is like for humans, for example, we secrete the nitrogen in the form of urea. Okay, so in this case, there are enzymes which are in the soil which are called as ureases, which will break down this urea into ammonia. So that is how ammonia goes back into the environment. So that is the second leg of the process. The third leg is called as the nitrification process. So when you go back to that slide, which I have shown you as an overview, you have a stretch where you see that anytime when you make nitrates from ammonia, this process is called as nitrification process. Okay, now what is so important about nitrate? Now, normally the nitrate does not accumulate in the natural undisturbed ecosystems. So the natural ecosystems don't have excess of ammonia either. So there is always a delicate balance. However, in agricultural systems, due to man-made activities, nitrification becomes important as excess of fertilizers are used. Or also in other managed systems, in septic tanks, etc., lot of nitrate becomes available, as a result of which there is too much of nitrate which accumulates in the soil. Okay, so because nitrate is an anion, it's very mobile in the soil systems, which also have an overall net negative charge. Therefore, nitrate can move easily with water, and this results in nitrate leaching into groundwater and surface waters. Okay, and there are several health concerns related to high level of nitrate, because if there is excess of nitrate, it can cause methemoglobinemia, which is a disease, and if excess of nitrate occurs into the biological systems, it is extremely toxic, or it can lead to formation of nitrosoamines, which are carcinogenic. High levels of nitrate in surface water can also lead to eutrophication. That means supplying excess of nutrients in the water bodies. And there can be anaerobic oxidation of ammonia into nitrate, etc. Okay, so in other words, that this nitrification process becomes of concern. Why? Because of man-made activities. If there is too much of accumulation of nitrate, then it has disadvantages. So is this point clear?
Yes, ma'am.
Yes, ma'am.
All right, then in that leg, we had the nitrate assimilation. That is, how does, very quickly, I will take you back. So, you see down here, nitrate assimilation. How does nitrate get converted into ammonium? Because these processes are constantly occurring in nature. So how does it occur? So just remember a simple reaction where you have, for example, the nitrate, which will combine with hydrogen, and these are special group of microorganisms which can convert nitrate into ammonia. The denitrification. So by denitrification, what we mean is the conversion of nitrate into nitrogen gas, which is the last leg of the process. Okay, and how does it occur? It is through several microbial activities that this occurs. So very quickly, what is shown here, for example, is that nitrate, for example, is taken up. So this is the cytoplasm, which is inside of the cell, and this is the periplasmic space. Okay, so nitrate, which is taken up, of course, nitrate can be assimilated also inside the cell, but then this nitrate, there are microorganisms which can convert this nitrate into nitrite. Okay, and many of these enzyme activities are listed down below. So you have nitrate reductase, which converts nitrate first into nitrite, and then the nitrite gets converted by, for example, nitrite reductase into nitric oxide. Okay, then there are enzymes which are nitric oxide reductase. So they will convert it into N2O, then N2O gets converted into nitrogen. All these processes may not have to be catalyzed by a single bacterium, but it is a group of microorganisms which together in the environment are responsible for converting nitrate into nitrogen. Okay, because it's not necessary that all the organisms will have all the enzymes. So it is always a consortium of microorganisms which are there. What is the practical application of it? As I said, that when you have excess of nitrate, which for whatever reason is coming into the wastewater bodies, in the previous slide, I had explained that how nitrate can actually build up in this system. Okay, so you would like to recover. Today, the thrust is that this nitrate doesn't have to be assembled by bacteria and end up just as a stagnant biomass, but you would like to recover nitrogen. You would like to throw it back into the environment as a dinitrogen gas. So in the wastewater treatment plant, nitrification and denitrifications are being integrated so that the nitrogen can be recovered from nitrogen containing compounds, and the rest of the material, for example, can be treated. It can be mineralized into carbon dioxide and water. Okay, so having known what is the biochemistry of these reactions, today there are several wastewater treatment plants which integrate denitrification and nitrification as a part of the treatment scheme. So this is something which I would like you to, keep in mind. So is this clear to everybody?
Yes, ma'am.
Okay. So when I actually come to recovery of energy from waste materials, okay, or recovery of these precious elements like nitrogen, you want to put it back into the environment. All right, that is also recovery of an important nutrient like nitrogen. We will again visit these processes like nitrification and denitrification because it's like a resource recovery. Now, finally, I will just very briefly cover with you sulfur cycle, and there's just one slide, 2 slides to explain that. And now sulfur is also extremely important because it is a constituent of all the sulfur-containing amino acid. Cysteine and threonine are 2 sulfur-containing amino acids which are a part of all proteins, and they are responsible for making the disulfide bridge in the protein. They are also responsible for allowing the proteins to fold properly inside the environment of the cell. This is the 10th most abundant element in the Earth's crust, and it makes up 1% of the biological organisms, predominantly in the form of proteins and other sulfur-containing coenzymes, etc. Now, it's not a limiting nutrient, except in very intensive agricultural practices. By intensive agricultural practices, that means where you are adding too many nitrogen fertilizers and you set up a target that I need to have so much of harvest per hectare of land. Okay, when there is a targeted agricultural, that time sulfur could become limiting and you may have to supply sulfur from outside. Okay, now sulfur is required for synthesis of amino acids, vitamins, hormones, and coenzymes, etc. And it is important as it forms a part of acid rain, acid mine drainage, corrosion of concrete, corrosion of metal, etc. All of these, they lead to sulfur buildup in the environment. Okay. Now let's briefly look at what are the sulfur reservoirs. So in the atmosphere, sulfur is present as either sulfur dioxide or H2S gas. Okay, now where do these gases come from? They come through the volcanic activity in the Earth's core and metric tons of sulfur, so many metric tons. It's a small reservoir of sulfur and it is actively recycled. In the ocean, you do have sulfate ions, which is a large reservoir, but then it doesn't get recycled as much. On the land, again, in the Earth's crust, we have ores like pyrite or gypsum, but they are not readily accessible. It's only the living matter through which the sulfur can be recycled back. Okay, so what does it also mean? It means that we need to understand what kind of microbial activities are occurring, which are responsible for releasing this sulfur back into the atmosphere. Okay, so that needs to be understood. So now, very briefly, in this thing, what I just want you to look at, for example, are first of all, that the microorganisms are going to take up sulfur generally in the form of sulfate ions. Okay, just like nitrogen is taken up in the form of nitrate or ammonium ions, similarly, the sulfur is taken up as sulfate ion. There are photosynthetic bacteria. Okay, there are photosynthetic bacteria which take up H2S. All right, and they only take up H2S in a manner so that the hydrogen is taken up and it is used to reduce carbon dioxide into carbohydrate, and then the remaining sulfur is released as elemental sulfur. Just as the higher forms of photosynthetic life, when they take up water from outside, they release molecular oxygen during photosynthesis. That is because they take H2O, hydrogen is assembled into the carbohydrates, and oxygen is released as molecular oxygen. But there are anaerobic bacteria which are photosynthetic, which take up H2S, and they release elemental sulfur. Like it shows here, phototrophic oxidation, it occurs, elemental sulfur deposits are there, or sulfide oxidation can also occur to give rise to elemental sulfur. Okay, now in this process, so when sulfate is taken up inside the cell, either you have something which is called as assimilatory sulfate reduction, or it is dissimilatory sulfate reduction. Okay, so please try to understand what this slide is all about. Of course, I'll give you the reading material also for this, but what you need to understand, therefore, is that, sulfur dioxide, the principal source of sulfate which occurs in the biological systems, is only in the form of either sulfate ions like calcium sulfate, or you can have sodium sulfate or ammonium sulfate as a salt when you are giving to a microbial culture in a shake flask. It takes up that sulfur. Of course, ammonium sulfate can serve as a source of nitrogen. It can also serve as a source of sulfur when sulfate ions are released. Okay, or it can be taken up as H2S. So in this sulfur cycle, there are only two things which I'm going to talk about. One is the assimilatory sulfate reduction. The other is the dissimilatory sulfate reduction. So are there any questions so far? Do you understand, what's the significance of the sulfur cycle?
Yes, ma'am.
Anybody else in the class, do you understand why sulfur cycle is important to know? Just like any other elemental cycle.
I had one doubt where you mentioned that in rigorous agricultural activities this is used. So I'm not clear with that point.
Okay, see, because, for example, it is not a limiting nutrient. In other words, if on a farmland, for example, that sulfur doesn't have to be supplied from outside under normal agricultural practices, but under managed agricultural practices, where, for example, when people want to increase the photosynthetic yield, they plant C4 plants. And I had told you that C4 plants, like all these grasses and all that, they are all C4 plants. All right, that is because you want to increase the photosynthetic yield, the amount of CO2 which you capture from the outside into biomass, you want to increase, and then you have C4 plants. Okay, now there sulfur can become a limiting nutrient. Then sulfur in the form of sulfates, etc., or when you are supplying nitrogen, you may want to give ammonium sulfate, or excess of ammonium sulfate may have to be given from outside into the soil. So under managed agricultural practices, where you have a target to increase the yield, then extra sulfur may have to be supplied from outside. Okay, because gypsum is also supplied in the farm practices, where excess of sulfates are provided to them. Okay, that's what we mean here, because it's an important, although it is classified as a micro element, it's not a macro element. it's a microelement which is required to be put into amino acids, vitamins, certain hormones, coenzymes, etc. Like coenzyme A, for example, also has sulfur. Okay, and because of mining activities, it is a part of acid rain because sulfur dioxide, which comes as a consequence of fossil fuel burning, combines with water and it makes sulfuric acid, which falls onto ground, and that is called as acid rain. So that becomes a problem because it can damage the soil, it can damage crops and other things, etc. Okay. So now in that sulfur cycle, there are two things which are listed here, which are, they look complicated, but they are not. Okay. So first we look at dissimilatory pathway. Okay. Now any dissimilatory pathway means that if you are going to take up sulfate ions from outside the cells, you are not going to assimilate them into amino acids or into sulfur containing compounds in the cell. Whereas in the assimilatory pathway, if you take up sulfate from outside, okay, it will be incorporated into cysteine, then it becomes a part of amino acids and biomass. So that's one thing which I want you to see. Okay, so in the dissimilatory pathway, when the sulfate is taken up, it is not assimilated into an amino acid, but it will end up as sulfide or as H2S, and it will be released into the soil. So it is not incorporated at all. So then why do you really want to take it up if you don't want to accumulate? It is because there are sulfate oxidizing bacteria, which don't take up molecular oxygen as the terminal electron acceptor during respiration. Because normally what do the bacteria do? They break down carbohydrates. The carbon is let off as carbon dioxide. The hydrogens are collected in the form of NADH. And all of them will get into the electron transport chain where oxygen will be the terminal electron acceptor. In these bacteria, which are called as the sulfate oxidizing bacteria, they don't take molecular oxygen because they live in anaerobic environment. So they take sulfate from outside and, ATP. So they convert it first into APS, adenosine 5' phosphosulfate, energy dependent process. Then they accept these electrons, which are coming actually through oxidation of these NADH, etc. And they make sulfide. They again accept electrons, and eventually sulfide is released as H2S into the environment. So they have not assembled it, but they have only taken sulfate as an electron acceptor in the oxidation process. Such a pathway is called as the dissimilatory pathway. So is that clear to everybody?
Yes, ma'am.
All right. What happens in the assimilatory pathway is that when sulfate gets assembled into cysteine, an amino acid, and then during protein synthesis, cysteine will be taken up into proteins. You are assimilating this. So what are the steps there? They are not very difficult to remember. Again, the same enzyme, which is ATP sulfurylase, activates sulfur first into adenosine 5' sulfate. Another molecule of ATP comes in to activate it into this. So it has two phosphates now. Okay, one donated from this ATP and the other donated from this ATP. Okay, and then thioredoxin comes and it makes a sulfide, which gets converted into sulfide, and then one of the enzymes will pick it up, add it to something else, and make cysteine amino acid. Okay, so that is principally the difference between dissimilatory and assimilatory. So the objective here is to be able to return it, the sulfate which you have taken up inside, you're again returning it back into the environment in the form of H2S. So it is an important part of sulfur pathway. What you have taken up, you are disassembling it, returning it back into the environment. Okay, so that's how sulfur is returned back. So please, these are the only cycles I'm going to be covering. And now we finished with the biogeochemical cycling. And what I'm going to do is I'm going to switch to now the next topic, which is going to be phytoremediation. All right. So have you been able to see this, the PPT, lecture 15? Do you see the first slide?
Yes, ma'am. We can
Okay.
see that.
All right.
Yes.
So now we are finished with the biogeochemical cycling, and we are starting with phytoremediation and microbial remediation, as the name suggests. Okay. Now we are going to be looking at, we have looked at a lot of this thing. Now we are just going to look at also technologies. That is how by phytoremediation, what we mean is that plants can be used in order to remove the pollutants from the environment. So that technology is called as phytoremediation, or certain microbes, based on the kind of biochemical activities that they possess, we can use them for remediation purpose, for removal of pollutants. So therefore, what is phytoremediation? Okay, so phytoremediation is a process whereby, and it's a very, very big, we are not doing it so much in India, but in countries like China, for example, they have field trials where, of course, they have a lot of land also. It's a very, very huge country where the soils which are contaminated with heavy metal ions or organic pollutants, etc., they plant trees in them, and over a period of 3-4 years, all of these metals, etc., which were in the soil, they are just removed by these plants. So let's imagine that if you have large agricultural land or some land where some mining activity has occurred, which has become contaminated with these metal ions, etc., you just plant some trees in there, and in 2 years' time, all these metals, organic pollutants which are in the soil are taken up by these plants. Okay, so then you just take these plants and put them somewhere else. So the soil which you were dealing with is already been remediated. The metal ions have been taken up by the plants, organic pollutants have been taken up by the plants. You take these plants and then you just incinerate them, or you take them elsewhere. Okay, so that can be applied on a large scale also. Yeah.
From where these heavy metal ions are coming?
Well, because of mining activity. So imagine that, there is a mine somewhere here, and the soil which is next to the mine is contaminated with heavy metals, or where you have the painting industry. Chromium is heavily used in the paints. Okay, so you have, for example, in all these industries, when the water bodies, they will all these pollutants, etc., chromium, etc., is not treated in there. It is just released into the water body. So through that, it can move into surface waters, it can move into soil. So large areas of soil get contaminated with heavy metals. So it's not like where the residential area is, but where mining activities are going on, or where, for example, when you do this oil drilling. Okay, so again, those areas are contaminated with these naphthalene and whatever hydrocarbons which are coming out there. The soils are contaminated with hydrocarbons there. Okay, so is that clear?
Yes.
As to what is the source of these heavy metals? So in other words, when you use the term phytoremediation, what you say is that involvement of plants for correction of soil, for removing the pollutant. Now, essentially, there are 4 different processes which are followed in plants. Okay, one plant could be carrying out one thing, other plant could be carrying out something. So there are so many trees or small plants which have been identified, which are capable of carrying out remediation. First process is called as phytostabilization. What is phytostabilization? So there are 4 things which are listed here, and I'm going to explain it to you. So these processes are either phytostabilization or phytoextraction, or phytostimulation or rhizodegradation, and phytodegradation or phytotransformation. 4 different mechanisms. So what is the meaning of phytostabilization? So in this process, it reduces the mobility and the bioavailability of metals and organic pollutants in the soil by preventing their migration in the soil and groundwater, or their entry into the food web. So what happens is that many of these metal ions, like you see in this picture, what is called as phytostabilization, is that the roots, etc., they are going to secrete certain organic compounds or something else in their cell wall, which is capable of binding this metal ions. So MT stands for either metal or metalloids. Okay, so what happens is that they are taken up and they will remain adsorbed on the surface of the soil. So these metal ions, in other words, are stabilized. They are not transported across the plant at all, but they are stabilized. They kind of bind them here. So in the free soil, all of these metals don't exist anymore, because they are taken up by these by the root cells in the outside of the root. For example, there are certain kind of materials which are produced. I will discuss it with you when we take it up a little bit in detail. Okay, so there are substances which are secreted in the outermost cells which can bind to metals or to organic compounds. So such a process is called as stabilization, because all the metals and everything are stabilized in the soil. They are not allowed to move, and they are not allowed to go through the plasma membrane, etc., into the rest of the plants, etc. They are just immobilized here. The second process is called as phytoextraction. Here, what they are doing, like you see in phytoextraction, okay, again, there are all these metals will enter inside the soil, but then through the transport systems which are located in the trunk, okay, these are allowed to accumulate and come up even up to the leaves. Okay, so in other words, the plants can now at this point, they can take up a lot more metal ions because they are able to mobilize them. They are able to store them in their leaves also. So that is also a kind of a remediation. So imagine a tree is so large, okay, and so many leaves and so many branches and everything. So they can build up a lot of concentration of these metals. That process is called as phytoextraction. So the third process is called as phytostimulation or rhizodegradation. So what it means that here the roots, yeah.
In case of phytostabilization, like we will have to plant these trees there, and they will fix these metals there. But
Yeah.
the metals are not being removed from there. So again, then how will that land will be used? There is no use of that land. So like why we are going to plant the trees, and is there any reason?
Because these metals, they bind to the cell surface. They become adsorbed on the root surface. When I show you a practical case, you will see that when you measure the amount of metal in the root, you will find that their concentration is gone up. And the outermost cells of the root, you will see that many of these will land up in there. There are carbohydrates, there are polymers which are made, which are able to bind to the metal ions. Okay, so they are removed from the soil, but they will enter and they will be deposited on the cells of the root. So when you take this plant out, you're also removing all these metals from there.
Okay, ma'am. Okay.
Okay. Now in the third process, which is called as phytostimulation, so or rhizodegradation, here the roots are basically used to stimulate the growth of bacteria and fungi on their surface. And so what do they do? The roots actually secrete out things like organic acids, sugars, amino acids, phenolics, certain enzymes, etc. And all of these are able to promote the growth of certain bacteria, which in turn are able to degrade all of these organic pollutants, or they are able to bind these metals. Okay, so that's why it's called as phytostimulation or rhizodegradation. All right, so they are going to stimulate the growth of these bacteria which are in the soil, which in turn are capable of binding the metal ions as well as breaking down organic compounds. That is called as rhizodegradation. And lastly, you have phytodegradation or phytotransformation, in which the plants themselves, the roots are going to make certain enzymes. So they are not polymeric materials, but they are capable of making enzymes like phenolic oxidases, lacases, peroxidases, etc., which are able to mineralize many of these organic pollutants. Or, maybe, that those enzymes are not a part of metal remediation, but those enzymes are a part of organic contaminant remediations. Okay, so is this clear to everybody? So these are the four principal methods. So now, I'm because in any of these processes, what you will see that the first thing that has to occur is that the metals have to enter inside the cell. Even in phytoextraction, the metal needs to enter inside the cell. Okay, so the question is that how do, after all, if there is a cadmium contamination, if there is an arsenic contamination, the plants are not supposed to be taking up. They are not designed naturally to take up arsenic or cadmium. Okay, then what happens? How do they get inside the cell? So in order to understand that, okay, there are, for example, in this picture, so transporters like inorganic phosphate transporter. Okay, the job of this transporter, which is localized in the membrane of a root cell, is normally to take up inorganic phosphate, but it can also take up arsenic, which is the pentavalent form of arsenic. Okay, similarly, Nib family aquaporins are transporters which are capable of carrying water inside. Earlier you used to think that water is only going to be carried through diffusion. But no, for there are large pores which are capable of carrying out water inside the plant roots. Okay, and these are called as Nib family. What is Nib? It is nodulin 26-like intrinsic protein. You don't have to remember so much of detail, but only appreciate, for example, that there are proteins which are meant to take up aquaporin, but they can also serve to transport arsenic in the trivalent form inside the cell. Now, apart from transporters, the cells, the plant cells, okay, they also have efflux transporters. That is, there are transporters which are localized, for which the function may not be known, but it has been observed that it can also efflux this arsenic trivalent form outside. Okay, similarly, in the cell, there are zip, COPT, full forms not necessary for you to know, except that these are transporters which normally take up copper. Okay, because copper is an important element. Copper is a part of all cytochromes. All iron sulfur clusters which are in the plant cells, which are a part of oxidation reduction, they're also copper containing. So copper anyway is an essential nutrient. So there are transporters, but then if there is a soil which is heavily contaminated with copper, the same transporter can serve to take up that extra copper inside the cell. And then there are efflux transporters. So these are heavy metal ATPases which can efflux back many of the metal ions, or manganese or zinc, etc. So these are a whole lot of these things, okay, which can also serve to take up things like arsenic or cadmium and other things like that. And on this side, we have, for example, there are iron transporters which are located. So it is only to give you an appreciation, or like, for example, cadmium here. Okay, cadmium is transported inside, and it can, in excess, lead, nickel can also be transported. So the idea here is only to allow you to understand that the first step, which is in phytoremediation, is the uptake of these metal ions. So how do the plant cells have any system for taking it up? And the answer is yes, that there are many of these transporters which are already, because heavy metal ions are contaminants, but metals like iron, metals like copper, metals like manganese, zinc, they are a part of normal biological activity. Manganese is a part of so many enzymes as a coenzyme. So these metal transporters themselves can serve to take up various metal ions inside. Okay, so let me stop here, and then we will come back and we will finish up this in the next lecture. And Saturday we meet again, because Saturday we are following Monday timetable. So is that clear to everybody?
Yes, ma'am.